Today we will be discussing the link between mitochondrial respiratory reduction and heart failure. During the course of this presentation, several acronyms will be used, so I'd like to take a moment to define them for you. HF, heart failure. LV, left ventricle. LVAD, left ventricle assist device. NFLV, non-failing left ventricle. GM, glutamate malate. PM, pyruvate malate. PC, palmitocarnitine malate. ETC, electron transport chain. RCR, respiratory control ratio. Scientists and physicians have long theorized that heart failure is caused by reduction in mitochondrial respiration within cardiac tissue. A group of scientists at the Weill Cornell Medical College decided to test this theory. To do this, they gathered sample mitochondria from LV cells taken from patients undergoing surgery to treat heart failure. Researchers found 24 patients who were set to undergo LVAD. Of these 24, 4 of them were NFLV. These 4 functioned as the experiment comparison group. Specific patient demographics can be seen on the table displayed. During the research leading up to the experiment, the scientists determined that a key factor in previous experiments' lack of significant results was the long lag time between sampling and analysis. To solve this problem, the researchers developed a new sampling procedure in which cell samples were taken during surgery and immediately taken to an adjacent lab for mitochondria isolation. To isolate the mitochondria, the cells were placed in a high-speed centrifuge, and using protocols established by Fariza et al., the mitochondria were removed from the cellular material. Following isolation, the samples were placed in a high-resolution Ouroboros respirometer to assess oxygen consumption, which was the key variable in the study. The study used 4 substrates to simulate different cellular environments: PC, PM, GM, and succinate. To better understand the respiratory process, each test was broken into 4 stages. Stage 1 was measured with just the mitochondria in the respirometer. Stage 2 added a substrate to the mitochondria. Stage 3 added ADP. And Stage 4 was full ADP-limited respiration. In addition to testing mitochondrial respiration, the mRNA of each sample was isolated to determine gene expression. Functionality of the extracted mitochondria was analyzed by assessing the oxygen consumption in each of the 4 substrates. Mitochondria isolated from NFLV tissue had similar stage 3 and 4 respiration rate in all substrates as seen in figure. The results you see in the figure suggest that isolated mitochondria have greater respiratory coupling with GM, but overall have no distinct differences ex vivo. Looking at figure 2, you can see that mitochondria from HF patients were capable of oxidative phosphorylation in GM, PM, and PC. RCR values in all substrates were comparable to previous models. The figure data indicates that the isolated mitochondria possess a strong ETC capacity in multiple substrates. The data from NFLV and HF patients are compared in figure 3, which show non-significant differences between the two. As previously noted in our demographics table, it was also necessary to determine if other factors played a role in mitochondrial function. No difference was noted between long-term and short-term HF sufferers. This result could show adaptive change based on specific body ailments. The researchers set out to correct a fundamental error in our scientific understanding of the causes of human heart failure. The evidence they gathered show that mitochondria isolated from LV tissues of the human heart exhibit coupled respiration ex vivo. The most unique part of the experiment, when compared to previous studies, was the quick turnaround time the scientists were able to produce. The scientists determined that energy starvation in HF was not due to a failure of the cardiac mitochondria. This was clearly shown given that the isolated mitochondria were capable of normal function in the presence of several different substrates. This study is important, as it suggests that some other root cause of HF must be at work and not mitochondrial failure as previously thought. This concludes our presentation of the relationship between mitochondrial respiration and heart failure. On behalf of our group, we hope that you've learned something interesting. Have a nice day.